So I'm a resident artist, science artist at the Walter and Eliza Hall Institute of Medical Research in Melbourne. I've been there for 20 years, and I created the position for myself, essentially. I'm a cell biologist, microscopist by background. I've transitioned now, and I just create visualizations using high-end graphics, computer game technology, and Hollywood cinema effects, and so on. Diversity of project. The main outcome is that I do at the Institute is this web page here, which has all sorts of clips, animations of all sorts of different topics that are related to the Institute: diabetes, cancer, apoptosis, malaria, and so on. And the idea is we create very high-end, high-value animations that do not ever dumb the science down. They are there to represent directly the actual scientific data, but when it's up to the narrative that you put to the piece, the language, the verbal language that suits the audience that you're targeting. So we create these animations. They're typically around 3 minutes a piece each. The idea is this is not the finished product. We then hand it on to science journalists, producers of documentaries, curators of museums, and also lots of YouTube and online stuff as well. And that's what I'm going to do for my first 5 minutes. I'm just going to play one clip. This came out in June. But it's very typical of the nature of the work that we do. So hopefully it'll play. I'm on the network. Yeah. So this is really a booming field now, which is charismatic science presenters on YouTube doing little short clips about science and making it engaging. And the work I'm about to present is from my student from about 2-3 years ago. She's still with me now, but it's on epigenetics. And to give you a sense of the work, it took her 12 months to produce this 3 minutes of animation. Most of that time is in research, 6 months of research, then technique development, and then actual outcome. We've used this animation in many different places, but this is a fun one. So he had 1 million views within 2 weeks, and we have lots of these sorts of relationships with other people doing this sort of content. So hopefully we'll have sound. Excellent. We'll see how this goes.
Inside each one of your cells, there is 6 feet of DNA, made up of 6 billion letters of genetic code. Now, your DNA is split into 46 pieces, each 3 to 4 centimeters long, called chromosomes. Now, normally we think of chromosomes as looking like this, but they only take that form when a cell is ready to divide. So usually, DNA is just a wiggly thread within the nucleus. Now, if you can imagine, DNA is only about 2 nanometers wide, but a chromosome is centimeters long, so you would think that it would get tangled worse than the headphones in your bag. So the DNA is actually wrapped around proteins called histones. Now those histones have wiggly tails, which will come in handy, as we'll see in a moment. Your unique set of DNA first formed when 23 chromosomes from your mom mixed with 23 from your dad. Now 22 of those chromosomes from each parent form matching pairs, but the 23rd set is the sex chromosomes. So 2 X chromosomes make you female, and an X and a Y make you male. Now, since the male sex chromosomes are different, both can remain active for the rest of your life. But for females, one of the X chromosomes needs to be inactivated in order for proper development to occur. This happens when a female embryo is just 4 days old and consists of only 100 cells. Right now, in this cell, the X chromosome from dad and the one from mom are both active. But through a tiny molecular battle, one of the X chromosomes wins and remains active, while the other is inactivated. This is done by packing the DNA closer together and making modifications to those dangly histone tails that signal this inactivation. New structural proteins are also added to bind everything closer together. And finally, methyl groups, these tiny little molecular markers, are added to the DNA to basically signal to the cell that this DNA shouldn't be read. So all of this together makes the DNA very difficult to access for the molecular machinery that would harness the code in this DNA. It is switched off. This DNA is silenced. In contrast, the active X chromosome DNA is more spread out. This allows better access to the genes on the chromosome. Histones can be slid along the DNA or removed entirely. and the histone tails have a different modification signaling this DNA is active. Now, all of this makes it possible for RNA polymerase to access and transcribe this DNA into messenger RNA, which then goes out into the cell and is used to make a protein. Now, what's surprising about X chromosome inactivation is that it seems kind of random which X chromosome wins. I mean, in some cells, dad's X chromosome wins, and in others, mom's X chromosome wins. So this 100-cell embryo ends up with a mixture of active X chromosomes. But from this point forward, as these cells divide, they maintain the active X chromosome that they had inside. So all of the cells with dad's active X chromosome give rise to further cells with dad's active X chromosome. And this continues on into adulthood. So if you could look at a woman's skin and see which X chromosome has been inactivated, you would see a stripy pattern, which shows the growth and migration of all of these first 100 cells when the embryo was just 4 days old. Now, of course, you can't actually see that in humans, but you can see this with calico cats. And that's because the gene for coat color is actually on the X chromosome. So just by looking at the pattern of her spots here, her dark and light spots, you can see where her mom or dad's X chromosome has been inactivated. And this also shows us that only female cats can be calico cats. And that's because, well, only female cats can inherit 2 X chromosomes with 2 different color genes. Now, this is just one really cool example of epigenetics. But epigenetics doesn't normally work on one whole chromosome. In fact, it's at play in all of your chromosomes, turning on and off your genes. For example, it's epigenetics which makes a pancreatic cell capable of producing insulin, because that gene is switched on there, but switched off everywhere else. What's more interesting is that it seems the behaviors you take can actually affect your epigenetics. And even weirder, perhaps the things that your parents or grandparents did can affect your epigenetics now, can affect your DNA. So you are not just a product of your genetic code. You're not just a product of your DNA. You are also a product of your epigenetics. And that is influenced by your behavior and the behavior of your ancestors.
Okay, that's the first outcome. That's really the bread and butter of what we do, is producing animations that get then repurposed to many different sorts of stories. Typically science programs, documentaries, and television shows. But we're certainly noticing a lot more of the surge of online programs like that. So switching to a completely different story. This is one do I want to do this one next? Yes, I do. This is a project over on the left here, and I'm playing it from my iPad here. And what it is a brand new interactive textbook of biology. It was a 2-year full-time production for me, and I had 3 staff overall. And there was we're a collaboration with Harvard. We had E.O. Wilson was he's a naturalist, the world's expert on ants, and a great writer. He was our lead author. We had a team at Harvard Medical School, and we were very intimately involved with Apple for technology. We had high school teachers around the US critiquing and so on. There's iTunes U courses, and so it goes on and on. If I had a day workshop, we could describe what we did in this book. I just want to play you a couple of quick examples. This just, we launched, I think, 2 months ago now, I guess. And it's for free on iBooks, and it's targeting primarily the curricula, high school kids, tens of millions of kids all around the world, but particularly in the US and California and so on. So I'm going to step into, it's broken over 7 books. I'm going to step into one of the books and just give you a sense of what this is like. So this is a chapter, and you can step through different chapters and look at. It's like it is a traditional textbook. We couldn't break the mold too much, but then the idea is you read the narrative, and it has figures as you'd expect in a textbook. But it also has things like this is not a still figure, but one that is an interactive figure. And this is really so you're following the narrative of this chapter, and you step into this figure. and this is a student playing with the iPad. The idea is they can then explore by touching and zooming. They can control scale, and they get a sense of the place and scale of this stuff. So here we are zooming into the skin. We keep on going, so there's sort of sweat glands and so on. We go through the epidermis, we go all the way down, and there's tissues and different levels of the epidermis, all the way down to a cell. And all of this done to scale. Everything is derived from data, but it gives the students a sense of scale, and this cell here then becomes the hub for the rest of the chapter. I don't have time to play too much, but I shall zoom out. Before I do that, I might go back over to here. Each of these figures is about a, credits. Each of these figures is about one of the different organelles. So there's a Golgi, which I don't have time to go into right now, but mitochondria is what I wanted to actually show you, because if we go to mitochondria in Google, this, yeah, of course. This is the way mitochondria are always represented. They have been for 50 years. This is the way it looks like in textbooks. There are these rigid jelly beans with the folds inside, very colorful. I mean, they don't have any color, so it's quite, you can use your artistic embellishment for that aspect. But time after time after time, they're short little jelly beans, rigid little things. That's completely not what they're like. And I wanted to address this. So this is just one little tiny taste before I move on to other projects, just to give you a little taste of, again, this is all derived actually from time-lapse microscopy of cells, different cell types, mitochondria looking different, but they're also very dynamic, and that's kind of what I want to convey.
The major function of the mitochondria is to provide the cell with energy in the form of the energy-rich molecule ATP. Mitochondria are dynamic and come in a variety of shapes and sizes, fusing and dividing to suit the changing needs of the cell.
And all of that sort of, I did that about 2 years ago. There were a number of papers in Nature and so on about the dynamics of mitochondria and how they're touching and changing, exchanging different compositions internally. And also what I was also representing, you have the microtubules in the background, which I will play you one more. This will give you a level of intensity. This is foot to the pedal to the metal, giving you a sense of the full-on biology we're going to give high school kids. But that had a number of dynamics where they're showing the endoplasmic reticulum and how it's involved with the fission of mitochondria as well. So there are a number of different sorts of forms of data that went into that visualization, even though that wasn't what it was about. Now this one is a super high level intensity, but for kids at this point of this book 2, and they've already seen a lot of these animations before, so I'm just apologizing for the amount of science you're about to receive, but hopefully it'll give you a sense of the nature of the material that we have created. So a lot of these shots are in different chapters: muscle structure, mitosis, and so on.
The cytoskeleton is a dynamic network of filamentous proteins that extends throughout the cytoplasm, forming the struts, cables, and girders that give the cell its shape, internal organization, and mechanical support for movement. There are several types of protein filament, each built from subunits that rapidly come together or break apart, allowing these elaborate structures to be assembled and dismantled wherever they're required by the cell. Microtubules are stiff, hollow tubes that anchor other organelles and serve as tracks that guide the movement of vesicles and other cell components. Actin are thin, flexible filaments that form cross-linked bundles and branching networks. Actin is particularly important for cell movement. They are central to the contractile engine in muscle cells and provide the mechanical force for cell movement at the plasma membrane.
Okay, so I mean, that was an intense amount of biology, probably too much, you'd argue, and I probably wouldn't disagree with you. We had a lot of constraints on this being file size, the movies couldn't be too long, and also the nature of what a textbook is. And what we were trying to challenge was the nature that there are $100, $150 printed book that had to be manufactured, warehoused, shipped to the students, and were 2 or 3 hours out of date usually before it reached the students. And what we were having was an online resource for free with all all the interactivity, all the videos. We have interviews with Nobel laureates. We have interviews with the scientists and how they made their discoveries. We have, it goes on and on. There's lots of experiments for the kids to do in their classrooms. The teacher can see, there's multiple choice questions. The teacher can see where students are spending time within the book, overview, as well as where they're getting stuck, so they can come back in the classroom. That's all provided, all connected together behind the scenes, as well as lectures and material appropriate for high school kids use in the classroom. So, yeah, so education, public education, high school education, and university education is a big area. It's been sort of the most important role that our animations are used for, and this was quite a big one. Actually, when am I going to wrap up? 7 minutes. All right, so I'm going to swap again and play you a whole another aspect of that I think is probably as equally important is my work in art. I was called upon, it's been about 10 years now, I've been doing lots of installations, art installations in museums and art galleries around the world, and I was reluctantly dragged into this many years ago by an arts festival, and what it turned out was, it was the most profoundly exciting and interesting thing, is that I'm reaching new audiences, people who turn up for art exhibitions. They're diverse, they have no background or interest in science, they never look to science for their entertainment or education, and they give work so much time, so much more than any other aspect that I've reached. Where I play an animation or a piece that goes for 20 minutes, they'll sit there and watch it once, they'll watch it again, they'll give it an hour, they'll contemplate it. What does the artist mean by this? What does this mean to me? These sorts of questions, which are fantastic, but also the conversations that go on. So anyway, this is one I did in February 22, is a major arts festival, like the Vid Festival in Sydney. And the idea is they do lots of projections on buildings, and it's free to the public, and there's too many people in the city, and it's all too crazy. But what they did was they gave me this famous room, which is the reading room in the State Library of Victoria. So it's 4 stories high, 35 meters across, dome, and so the people down the bottom there, to give you a sense of the daytime use of it. And they gave me this, and they said, here, just project stuff. And then you just project your animations. And I was looking at the room, and I thought, oh man, because you just go in, you just want to look at the room itself. It's like a cathedral you're in, and it's just too interesting. And I was looking at that dome, and it really was reminiscent of me of the outside coat of a virus. It looked like a virus, and I thought, hey, we could be in the virus, or we could be in the virus world, or something. And I have 6 weeks of production time, which most of my works are 6 to 12 months. So I had 6 weeks, and it was kind of mad, but I didn't have to do the projection component. But anyway, they gave me this room, and I decided to, if this is a virus, how big would it be? And it turns out, if it's a 30 nanometer virus, it becomes a 30 meter virus if you magnify it 1 billion times. So the idea was virus 1 billion times. I'm going to go ahead and play and talk over the top of it. The sound score was critical too. I'll come back to it later if you're interested. Actually, play. Here we go. So 8 human viruses, 2 minutes each, all of it derived from molecular X-ray crystallography. So you're in the virus world. We have the actual capsid of various viruses being projected on the dome. We had the genetic code of each virus being beamed on the bookshelf. So the entire, during each 2 minutes, you have the full genome of that particular virus. If it's an RNA virus, you have the RNA crawling around the walls in between the bookshelves. So they're transforming the nature of this library, a human library, into a genomic library. And these are all important human viruses. So this went one night from 7 p.m. to 7 a.m. It was at capacity. We had 22,000 people go through. That was 1,000 more than was allowed. There was an hour queue until 5 a.m. for people to come in and see this. And so this was HIV. Each virus had its own soundtrack and color palette. So very red and blood suggestive of HIV, and much more gloomy kind of a soundscape. And the idea was also, with each of the different soundtracks, we needed people to come in and we needed them to leave. So there are a couple of them that were quite aggressive and unpleasant, which is actually quite useful to get it, because the people sit there for 15 minutes and watch the thing. They might do it a second time, get around to the nasty one, go, I'm out of here, and leave. It was just what we wanted. Because people couldn't come in, so they left. Let's jump ahead. Rhinovirus, so the common cold. So of course I didn't not had these little factoids being on the bookshelves as well, little things to make people reflect. And the biggest, what was really fascinating to me, the most popular one, every time it came up was herpes. And it would talk about, in this room, back to Zoom here, in this room, 8 out of 10 people actually have the herpes cold sore virus. Most people are symptom-free, and those are the factoids I have on the wall. 2 out of 10 people in this room have the genital herpes. Most people are symptom-free. So those kind of factoids up on the walls too, they provoked all sorts of conversations, and it was really fascinating for me to sit amongst the general public and listen to how actually quite informed they are. You get, some drunk 20-something-year-olds would come in, and there'd be 8 or 10 of them, and they'd be Facebooking, whatever, and then there'd be one of them would know something about DNA or have an experience with papilloma or whatever it might have been, and it just generated a lot of really interesting conversations. Everything to scale 1 billion times was kind of the idea. And then influenza had the wonder of universe type sound score. There was, just to talk about the sound, it's all done by my buddy Frank Teahead, a long-time collaborator. It's good. It had a 4-second delay in the room, and we set up multi-channel, there's 8 channels up on the multiple floors, and so there are different parts of the sound components from different parts of the room. It was really quite interesting. Anyway, I will stop there because I'm out of time, I think. 5 minutes. I think we can do questions there now. Yep, all right. So, yeah.